this task, I have to go from test B to test A in chemistry. So there'll be an introduction, there'll be a pre-test, a break, self-study, break again, and a post-test and a survey. So I'll have 20 minutes to complete this test. You don't rush. If you cannot finish the test in time, move on to the next section. All right. Select the version that has been assigned to you. So I have to select biology version B, and then after 20 minutes I'll switch to the next slide. So before that, let's start a timer for 20 minutes, and here we go. Let's go back to the site. I have to go to test B. Yes, next. So this, I have to use this diagram to answer the following 6 questions. Alright, this is not my subject matter, but let's see. So label 6 represents an organelle that produces sugar molecules. What is it called? Label 6 is something like this. It produces sugar molecules. So sugar means energy. So I'll go with mitochondria, because that's what I know, that mitochondria is the powerhouse of the cell. Label 4 represents blank, which is necessary for. So label 4 is this vast area here. So this, I'm not sure, this is vesicle. Let's go with central vacuole, because I'm going with that because it's like in the central region. And maintaining turgidity, sorry. So I'll go with holding nutrients. I'm not sure about this answer at all. Label 10 is the internal layer of a substance that surrounds the cell, and this is called label 10. Label 10, cell wall is something which surrounds the cell. Right, I'll just go with cell wall. The main function of the smooth endoplasmic reticulum represented by label dash is dash. The main function of the smooth represented by label, let's see what label 2 and 9 represent first. So label 2 is this, and label 9, I have no clue what these things are. Alright, the main function 2, protein synthesis. Isn't DNA responsible for protein synthesis? But out of the two, one is responsible for protein synthesis, and 2 and 9. So I'll go with 2 as the protein synthesis one, and I'll call that, and 9 as the lipid synthesis. Alright, so it's not a multi. So I'll go with 2 with the protein synthesis. Label 5 is present in label 5. Alright. This again looks like a cell wall to me. Label 5 is present some animal cells only, plant cells only. It looks like cell wall to me. So could this be a no? This cannot. Nucleus. Alright, so I have no clue about this, so I'll go with prokaryotes only. The organelle labeled dash will be in higher number, or will be in higher numbers in muscle cells compared to fat cells. I have no idea what's responsible for muscle cells and what's, but I'll still look at what 1, 5, 8, 10 is. So 1 is this thing, 5 is the structure 5 is wall. 158 was it? So yeah, so 8 is, and 10 is, okay, so these two are two different structures that are shown here. So the question asks, this will be high in muscle cells compared to fat cells. 1, 5, 8. I'll go with 1, but I have no clue why. So yeah, this is done. The backbone of a polymer of nucleotide contains ribose sugar. What is this polymer? DNA, polypeptide. It says polymer of nucleotide contains ribose sugar. What is this polymer? I know this DNA is something called deoxyribonuclease, and this is ribonucleic as something. So I'll go with RNA, just because it sounds like ribose sugar. A DNA monomer is called a nucleotide, so I think it's like a smallest unit, and it's made up of 3 parts: a nitrogenous part, a ribose sugar. Alright, this something hints that a DNA is made up of 3 things. So I'll go with DNA. I'll just change this answer here. And a sulfate. Which part is correct? Alright. Deoxyribonucleic acid. I know this is called. Alright, I'll go with ribose sugar. I'll take this thing because I know this has something called ribonucleic acid. Why is RNA not the main carrier? RNA is single stranded, so it carries half the information. I know DNA are double helix structure, RNA is less stable. Uracil is toxic, and in high amounts it is lethal. RNA cannot form long chains. I believe this could be the reason, because DNA is a helix structure, double helix structure. Justify your answer for the previous question. Let's say does not form a helix structure, but still I have no clue about this thing. Okay, use this symbol to answer the following 7 questions. This is A, delta, epsilon, and T. According to the standardness of this molecule, what type of nucleic acid is? What type of nucleic acid is? This question is beyond my reach, because I have no idea what this symbol means in terms of DNAs or according to the base. Okay, let's stay with this question first. According to the standardness of this molecule, what type of nucleic acid, what type of nucleic acid is? What type of DNA? Okay, to the type of bases, strandedness. Okay, so I think there's something related to the strandedness of this molecule, the type of base in the molecule. I have no idea what the strandedness means, what the base means, but I'll go with, I even don't know what this TNTP means or NTP means, so I'll just guess. And this is paradoxical about this image. I don't even understand this image. According to your answer to question 11, what must be the final part? Phosphate, nitrogenous base, I guess amino acids. Okay, according to your answer to question 12, what must be the final part? Phosphate, nitrogen is base. What was the final part of the molecule? Phosphate. These two questions are similar. Right. Is there something which I'm missing here? So, according to your answer to question 12, what will be the final part of the molecule? Phosphate. Excuse me. Phosphate, nitrogen is base, and this is exactly the same question. Use the following information along with the previous to answer the following two questions. After testing, this nucleic acid is found to be the entirety of a thriving and successful alien's genome. Chargaff's rules still apply. A deoxyribose sugar is found in its backbone. What are delta and epsilon most likely to be? So it's in the genome. I know there is something called A and T, but I'm not sure what these are. Both A. I'll go with A and T because what conclusion can we from find? A genome that is comprised of single strands is sufficient as genome needs C and G. The type of sugar. This must. Why will be? I have no clue what Chargaff's rule is. I believe C and G are the correct answers. Reverse transcription is the inverse process to the ordinary central dogma of biology. It can be represented as, separate your answers. I have no clue what this sentence even means. The inverse of the process. Reverse transcription is the inverse process to the ordinary central dogma of biology. The inverse of the process described in the previous question is reverse transcription. Genesis, RNA synthesis, ribonucleogenesis, transcription. Presented as. So I have to give something which is the inverse of that. So it can be transcription because. Okay, so it can be transcription because we are asked about reverse transcription. Let's go with transcription. Which statement about protein is correct? A straight chain of amino acids is indicative of primary structure. A beta spiral is indicative of secondary structure. A protein bonding with another is indicative. Quaternary structure is completely impossible. Straight chain is indicative. Tertiary structure. I'll go with this protein bonding because I know that if I remember correctly that protein bonds are necessary for this thing. So let's go to the next section. Use this diagram to answer the following three questions. I have no clue what this diagram means. These are like U, A, C, G. A DNA template strand contains this sequence GTA, TTG, AAA, GCT, GTG. There's something 3 dash or something. What is the complementary strand of mRNA? I'll go with some random thing. I have no clue what this even means. What is most likely reason that the sequence of the question has been returned in reverse order from the answer? Order from the answers. Okay, this tells me that was it supposed to be in the reverse order? GTG, but there's nothing. The template strand is at 3 2 and the mRNA is synthesized 5 dash to 3 dash. I'll go with C because this sounds more reasonable to me. What protein sequence does this mRNA code for? Separate each protein. I don't even know this. point mutation has occurred, but the polypeptide is the same. What is this mutation called? I know mutation is change in the DNA. About a point mutation has occurred, but the polypeptide is the same. What is this mutation called? Miss, I'm just guessing. A point mutation occurs in a separate polypeptide. The second one interacts with the first one in an epistatic manner and causes the original polypeptide to become truncated. What type of mutation is this? The second called. So there is a mutation which happened in a separate polypeptide. This is like new, different from the first one. The second one interacts with the first one in an epistatic manner and causes the polypeptide to become truncated. I'll go with D, but it's completely random. Okay, let's stop the timer and view the scores. I believe, yeah, so true. Okay, label 6. Okay, this is green. Okay, chloroplast is responsible for the colors. I believe all my... Okay, central vacuole. It's there's something called membrane. There's something called cell wall. There's something called membrane. Yes, the main function of the smooth endoplastic reticulum is represented by... Let's see what line was again. I've forgotten that. Alright, so this is for lipid synthesis. Some protein. Okay. Being higher number. Alright, this one was correct. Great. The backbone of a polymer is RNA. Okay, the molecule contains ribose. I should have gone with RNA. DNA is called a nucleotide, so it's, but this already has a nitrogenous base and ribose and sulfur. Which part? Oh, right. So nitrogenous base is the correct part, not ribose. RNA is less stable than DNA. RNA has one more hydroxyl group as its. According to the strandedness of this molecule, what type of nucleic acid it is? I didn't even know what this was. Bases. The single-stranded nature indicates RNA. What are the types? According to your question, what is the final ribose? I had no clue. Alright, this was another guess. This worked out. Organism C and G. So this was more like thing. So I had no clue what the image states. Although this was the C option made most sense to me, so I went with this. And I had no clue, and I don't even understand what is it. Histamine? No, sorry. Point mutation has occurred. Alright, so it's called silent. Alright, so it's okay. So I believe I have hardly answered 3 or 4 questions correctly. Sorry. So I went to B. This part is 10. No, sorry, this is not the correct slide. I have to go. Yeah, so I was here. I take a 5 minutes break. Right. So should I start a timer for 5? It's approximately 5 minutes. So now comes the self-study part. Please ensure you're talking through your thought and learning process. For the first 30 minutes of your study session, copy and paste each question you got wrong, that's most of the questions, into the Google and append -AI or -AI. There is a question where you must input an image and to attempt to grasp the key concept of the question and search that instead. If Alright, so I don't have to insert any image. If you finish before 30 minutes, continue reviewing. If you do not get through everything by the end of 30 minutes, move on to the next section anyway. Alright, so let's start a timer for 30 minutes. I'll just open my older tab. So I have to start a timer for, yes, so here it is. I'll just minimize this one and go back here. I'll just keep both the screens side by side. Let's see if I can do that. Alright, so this is alright, so this is the part where, so I don't have to put the images there, so I'll just ask this question. So can I copy this? Okay, let's try keeping them both side to side. This is not working. Yeah, so this is how it goes here. Okay. Yeah, so now it's better. I just have to write this and I have to ensure that I put -AI. I'll just reframe the question, the part in this cell. So we have not been specified what should I include the options or not, so I'll just keep that as it is and I'll just write -AI. Alright, so alright, so this is suggesting chloroplast, not mitochondria, so I'll ask Wikipedia. That's one of the trusted source. So chloroplast is a type of organelle known as plastid, okay, that conducts by photosynthesis. Alright, so this is the green part that's responsible for the photosynthesis, mostly in plant and algal cells. They have high concentration of chlorophyll, hence green, pigments which capture the energy from the sunlight and convert it to chemical energy and release oxygen. That's then used is getting converted to make sugar and other molecules. So let's see what this structure of, right, this is actually looking like that. It has other things in it too. So there is some outer membrane, inner membrane, cranum. I have no clue what this is. The chemical energy then is used to convert sugar and organic molecules and other organic molecules from carbon dioxide is in a process called Calvin cycle. Chloroplasts carry out a number of other functions, including fatty acid synthesis. So this is responsible for sugar production. That's what was asked in the question. It's responsible for the green color in the plants. That's something which chloroplast and so this is present in both plants and algae. They're highly dynamic and are moving around the cells. Their behavior depends on the environmental factors. They cannot be made anew by the plant cell and must be inherited by each daughter cell. So it's something which is genetically passed on. That's what I'm understanding from here. Chloroplast evolved from an ancient cyanobacterium that was engulfed by an early eukaryotic cells. Because of their endosymbiotic origins, chloroplasts, like mitochondria, contain their own DNA separate from the cell nucleus. Alright, that's interesting. With one exception, the amebioid Pollinelia, all chloroplasts can be traced back to a single endosymbiotic event. Despite this, chloroplasts can be found in extremely diverse organisms. Alright, so this is interesting. I'll just stay till here because I know that I understood the point that they are the sugar molecule producing cells. So the next part asks for what this label 4 represents. Let me ask, what is central vacuole in plant cell and why is it necessary? Okay, I'm not sure what level of information it will provide, but let's go with this Cora, or no, let's go with the one provided by some university. I've lost that link. Yeah, this is vacuoles. So I'll just bring the diagram here. Okay, so this is the central vacuole. Vacuoles are membrane bound sacs. Okay, they have a membrane around them within the cytoplasm of a cell that functions in several different ways. In mature plant cells, vacuoles tend to be very large and are extremely important in providing structural support. Alright, and as serving functions as storage, waste disposal, protection, and growth. Many plant cells have a large central vacuole that typically takes up most of the room. Vacuoles in animal cells, however, tend to be much smaller and are commonly used for stored materials or to transport. Okay, this is a large thing which is more like a structural support, but it's a waste storage. It doesn't store nutrients. It's a waste storage. It functions as a waste storage thing. The central vacuole enclosed by membrane termed the tonoplast. An important and highly integrated component of plant membrane network system. This large vacuole slowly develops as the cell matures by fusion of smaller vacuoles. Okay, this is so the central vacuole also contains that. Among its role in plant cell functions, it stores salts, but that's not. Yeah, it's written here. The central vacuole stores salts, minerals, nutrients, proteins, pigments, helps in plant growth. So this thing can be correct. Okay, let's go forward now. It plays an important structural role, but yes, the main role is for the structural support, so that's more prevalent here. Under optimal conditions, the vacuoles are filled with water to the point that they exert a sufficient pressure against the cell wall. Alright, so then the turgidity or the structural integrity part is more important for the central vacuole. This helps in maintaining the structural integrity along with the support from the cells and enables the plant to grow much larger. The structural importance of the plant vacuole is related to its ability to control turgor pressure. Turgor pressure dictates the rigidity of the cell is composed with the difference between the osmotic inside and outside. Osmotic pressure is the pressure required to prevent the fluid diffusing through a semi-permeable separating two solutions contain. Alright, okay, this is interesting. The response of plant cells to water is a prime example. When a plant receives adequate amounts of water, the central vacuoles of its cells swells up as a liquid, creating a high level which helps maintain the structural integrity. In the absence of enough water, they shrink, the turgor pressure is reduced, comprising the plant's integrity. Okay, that's how the wilting takes place. The plant vacuoles are also important for their role in molecular degradation and storage. So this bigger part, in short, is the vacuole, and out of the two options, this maintaining turgidity or the structural property is important. Label 10 is the internal layer of a substance. So cell wall is, I think, the outer part. So the internal layer, I'll just go with this, called cell wall. Alright. The cell membrane. The cell membrane, and historically is a biological membrane that separates and protects the interior from the outside. It's a lipid bilayer consisting of phospholipids and glycolipids. Phospholipids and glycolipids. Eukaryotes and prokaryotes typically have sterols, such as cholesterol and animals, intercepted between them as walls, maintaining appropriate membrane fluidity. The membrane contains membrane proteins that span the membrane and serve as transporters. Alright. Controls the movement of substance. Alright. Being selectively permeable. Oh, I knew this part actually. It's selectively permeable to ions. In addition, cell membranes are involved in a variety of cellular processes such as adhesion, ion conductivity, including. Okay, let's read this line again. In addition, cell membranes are involved in a variety of cellular processes such as cell adhesion, ion conductivity, and cell signaling, and serve as the attachment surface for several extra cellular structures, including cell wall and the carbohydrate layer called the glycocalyx, as well as intracellular network of protein. This is too technical for me, but let's see how the cell wall looks like just for the remembrance part. Okay, and let's see how the cell wall looks like here. So I understood that it's present in both prokaryotes and eukaryotes. And let's open this image. No, this is not the whole cell structure. There's no image here. Let's go back and see if I have the image so I can. Show the image. How's that? Okay, let's try another website now. I just want to see nucleus. So I'll have to take this. So this is the cell wall. What's this 5 called? There's something which is 10. I don't know what 5 is. So I have no idea how to search for that. Let me guess, and let's write is cell wall. I hope this one is cell wall, present in some prokaryotes and some eukaryotes. Okay, cell wall. I'm not sure how to answer this, ask this, and understand this, but I have. Let's look for the cell and cell diagram. Just to have an idea what this is, so that I can ask the right question. Or layer before. Okay, as seen in a cell membrane is vital. Let's write this. No, but I asked it not to give me some AI answer. Still, it gave me. So I'm just taking that and I'm opening a new tab here. Lipid bilayer. Lipid bilayer present in some. Let's copy that. Just to finish up writing part now. So both prokaryote and eukaryotic cells have a plasma membrane, a phospholipid bilayer with a mediator separates the internal contents. Cell membranes. Maybe this might show the diagram is a thin polar membrane made of two lipid molecules. These membranes form a continuous barrier for organisms. Cell membranes of all organisms and viruses made of lipid. No, I'm not. I'm sorry. This part, I think it's very difficult to make. The model and this on to understand without actually putting the diagram. Alright, I'm leaving this. I'm still writing. Okay, this one is correct. I just want to have because this was my guesswork. Minus AI. Why does the muscle cell have more organelles? The organelle labels. This cells need more mitochondria because they have to really run. Okay. So the whole thing labeled one. This is the mitochondria. Oh, this two things actually look similar, but they are different. They are green and so. Okay, so this is also new for me. The backbone of a polymer of nucleotides contain. What is this polymer? This can be put as it is. What the backbone of a polymer of nucleotides contained. Backbone of polymer of nucleotides is ribose sugar. Alright. Should I give the options also here? So let's try. There's something. Okay. Nucleic acids are the polymer of. I'll go with this side now. Are the polymer of nucleotides? No, this is not correct. It's so difficult to get the end. Well, this polymer. Excuse me. Then polymers. No, this is. Okay, let's add something. Ribose. Structure at study.com. Let's go there. Polynucleotide structure, chain structure and overview. Right, this is a video. I don't have time for that. Okay, this might help me. Nutritional Medicine Center. DNA is deoxyribonucleic. I was right about that. It's a genomic material that contains So I'll just look for RNA descriptions to RNA. DNA methylation. It's too tough. Inference as a process that moderates dice a double strand. A double strand RNA specific and design protein coding RNA. I am not getting this answer anywhere. DNA monomer is called a nucleotide and made up of 3 parts. Which part is correct? Three parts. A DNA is called a nucleotide. The 3 parts of a nucleotide. Okay, let's go with Wikipedia. Nucleotides are organic molecules of a nitrogenous and a base, a pentose sugar. Okay, this is something called a pentose sugar. I had no idea about. And a phosphate, not a sulfate group. It's a phosphate group. They serve as monomeric units of nucleic acid, deoxyribonucleic and ribonucleic acid, both of which are essential molecules within all life forms. Nucleotides are obtained in the diet and also synthesized by the liver. Nutrients by the liver. Nucleotides are composed of 3 subunits: a nucleobase, a 5-carbon sugar. Okay, nucleotides are organic molecules, are composed of 3 subunit molecules. Okay, so there is a nucleobase, a 5-carbon, a ribose. So nucleotide is composed of something called ribose sugar, a deoxyribose, and a phosphate group. It's still a phosphate group. I'm not sure what this 5-carbon sugar or this pentose sugar is. The 4 nucleobases are this is adenine, cytosine, thymine, and I was right. Adenine, guanine. Nucleotides also play in metabolism. They provide chemical energy in the form of nucleoside, ATP, adenosine phosphate, GTP, and UTP. Uridine. Oh, that's the U. That was stand as well. Throughout the cell, for many cellular, like amino acid, protein, and cell membrane, moving the cell and cell parts. So there is a nitrogenous base. There is no ribose sugar, and there is a phosphate base. It's rather than a ribose sugar. There is a pentose sugar. Why is RNA not? I think this might be answered pretty well here. I also want to know what this is. RNA is less stable. RNA is less stable. Okay, that's true. Sorry, this thing is blocked here. Let's ask Cora then. Well, RNA is genetic, not in plants or animals, but in some. Okay, this is wrong. This is not a reliable thing. This sounds. There will be some text. Okay, let's look at this. RNA functions as an enzyme and is therefore reactive and okay. Uracil presence in RNA is less stable. What is a U thing is that's present as compared to thymine. That's the difference between RNA and there is UTP instead of. I am not sure. Uracil is present. Being unstable, RNA mutates at a much faster. That's why RNA viruses have shorter lifespans. Oh, that's interesting. And they mutate and evolve very fast. Such rapid changes, of course, they are harmful to higher forms. But this reasoning was not given. They have one more. So this is said that U is present, uracil is present. Let's write. Let's find something else. I mean, it was a good thing, but not. Let's find this article here. So DNA has a base pair, helix. This helix. So it's a single one. So it has U. It has thymine. It has cytosine, adenine, and guanine. Oh, it's UCAG, TCAG. What's this hydroxyl group? Deoxyribonone, they are both responsible for reading genetic materials. They have linear polymers consisting of Sugar phosphates is a key difference. This is enable two molecules to work together and fulfill. DNA versus a compare function, sugar bases, something. Unusual types of RNA. Okay. Function, sugar base. Alright. Okay, this is the function. DNA replicates and stores genetic material. It's a blueprint for all genetic information. RNA converts the genetic material contained within the DNA to a format used to build proteins. It stores the genetic material. It converts the genetic information in the DNA to format used to a protein and moves it to ribosomal protein factory. DNA consists of two strands. RNA has one strand, but like DNA is made of nucleotide. RNA is shorter. Each nucleotide contains phosphate of 5 carbon and a nitrogen. Structure but only intermittently. Alright, DNA is much more longer. A chromosome is a single long DNA molecule which will be several centimeters and unraveled. Its variable length, it's shorter because they're unstable. There is deoxyribose which contains sugar. Deoxyribose which contains one less hydroxyl group than RNA. RNA contains ribose sugar molecules without the hydroxyl modification. So this is not the only it's less stable. The base in DNA, so this is not mentioned that it's a reason for the instability. ATG, and this has a U. Adenine, guanine, and cytosine. The thymine is the only adenine 8T. So base pairs are AT, CG, AC. I have no idea. Okay, A and T, A and U. So and C and G, C and G. I think that's the best way I can remember. DNA is found with a small amount of present mitochondria. Okay, so this nucleus of the cell. RNA forms in nucleolus and then moves to special regions of cytosol. I don't know what nucleolus is. The subunit. Due to its deoxyribose sugar, which contains one less oxygen containing hydroxyl group. Okay, now it's there. DNA is more stable than RNA. It has one less oxygen containing group, more reactive and is not stable in alkaline conditions. It's subject to attack. RNA is more resistant to damage. Its volume. Yeah, so that I knew. So I only have 3 minutes left. I'm not allowed to put image. So I'll just add something A. Epsilon and T. AT. Now let's copy this question. I really want to know what this delta and epsilon is. So DNA polymers delta. This is so difficult to understand this. Let's go with the science diagram itself. Poly delta. No, I can't find without. The single-stranded nature indicates RNA, while the type of bases indicate DNA. According to your answer, I don't even understand. After testing, this nucleic acid is found to be entirety sugar. Okay, so this is an image. A and T. Okay, so this is AT. That is composed of single-stranded is as sufficient. Reverse transcription is a inverse process. Just because I have very less time to modify reverse flow of information. There is a central drop of a molecule. Okay, central drop. Let's read this thing. RNA to DNA. Resolve the question regarding the picture of understanding. A reverse transcription process where RNA is converted into DNA. Complementary DNA. Okay, this is the opposite of the usual transcription process. Okay, identify the enzyme. The enzyme responsible for reverse is called reverse transcriptase. This enzyme is an RNA dependent DNA polymerase, meaning it synthesizes DNA using an RNA template. In reverse, the sequence starts with which is then converted into cDNA. This cDNA can be transcribed back to RNA, and finally the RNA can be translated to protein. RNA to reverse cDNA to RNA transcription, RNA translation. Okay, this is called translation. The inverse is called transcription. That's I know. A protein straight chain is indicative of primary. Okay, so time's up. Alright, so this was the least I could do. I'll just. Oh, sorry. Okay. Now it is my document. Oh, sorry. Yeah, this is my document. Let's see what I have to do next. I have given 30 minutes. I was not able to finish the whole review, so I'll just move to the next section anyway. Okay, now I have to give 15 minutes to these topics, and I have to search again by using this template minus AI. Animal cell diagram, plant cell diagram, function of cell, deoxy versus ribose, central dogma of biology and DNA. Do not try to finish all the. Even if you do not finish them all, we'll move on to the next section anyway. Alright, let's set a timer for 15 minutes now. Oh, sorry, this is. Let's remove that. I'll add a new tab and animal cell diagram. Okay. This nothing that's explaining that, so I think this is the best one I have, which is, so they're just showing me the pictures. I'll just choose one which resembles the one I had there. So let's try and open this. So this is the nucleus, the bigger part. This is vacuole. The cytoplasm, the liquid. So the vacuole layer is small. Yeah, so that's what the rigidity of the surface. This is the nucleus. Oh, this inside the nucleus we have nucleolus. Then I have endoplasmic reticulum, smooth endoplasmic. Oh, this is rough. The dots, smooth centrioles, Golgi apparatus. The ribosomes. I know what ribosomes are, but I'm not remembering it. Lysosomes, mitochondria. I know that. Peroxisome, microtubules, cytoplasm, cell membrane. Alright. So let's compare this with the plant cell diagram. Best one I can get is again from the same side. So I have this one. I just open this. This will help me compare. Can I? Yes, this is better. just sorry, just move this here. So there are some things which are not there, and there's a cell wall, a meloplast and chloroplast. This is a vacuole, I don't know. This is nucleolus, nucleus. This is something rough, endoplastic reticulum, smooth. Vacuole is smaller here, larger here. Peroxisome is there. Golgi apparatus is similar there. Cell membrane is there, and the liquid cytoplasm is there. Ribosomes are there, and lysosomes. Other than that, the only major difference is these three and centrioles. Alright, let's go with the third one. I'll go with this side here. Organelle, I hope I'm pronouncing this correct. Any of this specialized structure within a cell that performs organelle. The question, like, this cell's recycling center. I have to go with this side, I guess. A cell is a basic structure. The cellular components are called. The cell organelle includes both membrane and non-membrane bound organelles present. Okay, they coordinate and function efficiently for the normal functioning. A few of them functioning by providing shape and support, whereas some are involved in the locomotion and reproduction. There are various organelles without membrane. The cell ribosomes and cyto are non-membrane bound cell organelles. They are present in both in. Okay, but the things within the cells which do not have a membrane, they're present in both prokaryotic and eukaryotic. For example, the ribosomes, that is single membrane bound vacuole, lysosome, Golgi apparatus, endoplastic reticulum are single membrane bound. There are double membrane nucleus, mitochondria and chloroplast. So they are double membrane, and they are present only in a eukaryotic. Okay, the single membrane and double membranes are only found in eukaryotic cells. So I believe eukaryotic means cells without nucleus. They have no true nucleus and membrane. Right. So there are some cell organelles which are also present within the eukaryotic cell that do not have membranes. This is something I looked at before also, but let's go with that again. What's the difference between RNA and DNA? Let's go with Britannica now. Deoxy and ribonucleic acid and deoxyribonucleic acid are complex molecular structures that control all the hereditary characteristics of the cells. DNA is the master blueprint for life and constitutes the genetic material, and then uses DNA to code for the structure of protein synthesis, and it's also called the genetic material of certain viruses. There are several chemical differences between the two structures. As the name, deoxyribonucleic, DNA has a sugar known as deoxy, so it doesn't have this extra OH that I understood in the backbone. Ribonucleic acid has a slightly different sugar called ribose. Both are made of combinations of 4 nucleotides, which are special building block molecules. DNA is composed of this adenine, this T and U that differs, that I understood, adenine, guanine, cytosine and thymine. In RNA, the thymine is replaced by uracil. Other than that adenine, AGC. A and T, they form the block A and T, and U and T. The order and patterns form the genetic. DNA is a double structure, while RNA is a single strand. Alright. This is the only thing that's written here. Okay, that's interesting. Unlike most other organisms, chloroplasts and mitochondria have smaller chromosomes. What's the difference between deoxyribose versus sugar? Ribose sugar. Excuse me. Let's go with Cora this time. Oh, they have shown the structure. So deoxyribose is C5H10O4. Alright, so this has this extra OH which makes the ribose, which makes the RNA unstable. So they have the CH2OH, has one less oxygen atom compared to ribose, H10O4 and C5H10O5. Is normal with one oxygen atom attached to each carbon. Deoxyribose found in DNA is a modified lacking one oxygen. DNA and RNA are both crucial. DNA is located in the nucleus and RNA is located in the cytoplasm. Okay. DNA provides a genetic code, which in turn provides a genetic code to create amino acids, the building block of protein. Let's go with something more. This sounds like a chapter. Okay, this is not valid. Okay, I hope this is not a video. I don't want to watch a video. I just alright, this the difference in deoxyribose and ribose sugar is in the second carbon. I guess they're talking about the second, like the two carbons there. Okay, ribose has a chemical formula. Analyze the difference. The key difference is when oxygen atoms locate the position. The oxygen differentiate is located at the second carbon. Yeah, that's understood from the diagram. In ribose, the second carbon has OH group, and deoxy, the second carbon has H group. That is an atom. That's the only. What's the central dogma of molecular biology? Alright, so I'll just go with the first link that's provided here. It states molecular biology, but I hope it mentions the same thing. The central dogma of molecular biology deals with the flow of genetic information within a biological system. It is often stated that DNA makes RNA and RNA makes protein. Right. Although this is not its original meaning, it was first stated by Francis Crick. It states that once the information has passed into protein, it cannot get out again. In more detail, the transfer of information from nucleic acid to nucleic acid or from the nucleic acid to protein may be possible. The transfer from protein to protein or from protein to nucleic acid. Okay, the flow of information is unidirectional. So I can transfer an information from a nucleic acid to nucleic acid or to protein. But from protein to nucleic acid is not possible. Here means the precise determination of the sequence. I understood the DNA material, either of bases. He restated in Nature that central dogma deals with a detailed residue by residue transform. I didn't understand. States that such information cannot be transferred. Okay. The second version is popular and current. This is simplistic DNA to RNA, RNA to protein. The first molecular biology. Watson's version differ from Crick's. Because Watson get two-step DNA to RNA atom because of the central process as a central, while the dogma is stated remains valid today. Watson's version does not. Sorry. The biopolymers that comprises DNA, RNA are linear heteropolymers. I don't know what linear heteropolymers are. In the sense that DNA replication must occur if genetic material is to be provided for the progeny of any cell, whether somatic or reproductive. The copying from DNA to DNA is arguably is the fundamental step. A complex group of proteins called ribosome performs the replication from the parent strand to the complementary daughter strand. Transcription is a process by which the information contained in a section of DNA is replicated in form of a newly assembled piece of messenger. Enzymes facilitating are called RNA polymerase and transcription. In eukaryotic cells having a nucleus, the primary transcript is pre-mRNA. This is I'll just go for translation. The mature mRNA finds its way to ribosome where it gets translated in prokaryotic, which have no nuclear component. The process of translation may be linked together without layer. In eukaryotic, the site of transcription is usually separated from DNA to RNA. I think this is from RNA to protein or RNA to C DNA, something like that. Condones. I don't know. The ribosome reads the mRNA to codones with AUG. U is uracil or initiator. Reverse transit process from RNA to DNA. Oh, this is called reverse. Translation is different. On this way to this guy translated, which have no nuclear component. Transcription is DNA to RNA. I think from RNA to translation in which proteins are produced. Okay. The mRNA doesn't. Reverse translation. This is known to occur in the cases of retrovirus. In this case, this is interesting. This was just by which the genetic information gets transcribed into a new DNA. Okay, I'll just go with this. Okay, I think I'm out of time now. So I'll just stop. So I was not able. So I'll answer these practice questions. What part of cell contains the instruction for life? I'm not sure what Golgi apparatus is for. Not the cell wall. Cell wall is responsible for the flow. That's understood. I guess nucleus. Nucleus has DNA or nucleolus out of the two. The smooth and rough endoplasmic reticulum both synthesize a molecule. The smooth produces, I think smooth has RNA. I'm not sure about the other one. The rough, I guess nucleus is responsible. Choose a symbol that best corresponds to DNA. I'll just choose T. Sorry. Symbol U. I'll just write U here. That's interesting. The central dogma of biology is a synonym for the flow of information in a cell, from DNA to RNA to protein. This is in a eukaryote. This having nucleus, DNA is scattered throughout the cell. This is false, I guess. It's still in the nucleus. A pairs with T. Yeah. What is the effect of mutation that does not change? What is the effect? I was not able to go towards the mutation part. What is the effect of a mutation that does not change the amino acid? The effect of mutation, was there given some? Sorry, I just need to check. Oh, sorry, I just had to set a timer for 15. I'll just make it 14. I'll just make it 13 because I've already spent time on 2 the questions. I forgot. What is the effect of mutation that does not change the amino acids? I'll just leave that. The, there's another thing. The two main types of secondary structures are alpha. I'm not sure what primary and secondary structures are, but let's see what rhymes with them. Alpha spirals, beta sheets, alpha helices. Alpha spirals and sheets. I know helices. I'll go with helices and sheets. It's completely a guess from my side. What is the effect of a mutation that does not change the amino acid that the codon codes for? If the amino acid is not changed, I'll go with nothing. So yeah, so I have to space practice questions. I have done that. Should I look? Okay, this I still want to look for my score, so I'll just go. Yes. Oh, the smooth lipids and proteins, yeah, fats and okay. Okay. Oh, I didn't know that. I definitely didn't know that the symbols were not shown anywhere. The central dogma of biology is this is DNA pairs where what is the effect? No, this went well. But these two were completely guesses. But okay, so I need to take a 10 minutes break. Yeah, so anyway I have 10 minutes here. Okay, so I'll just keep a buffer of 1-2 minutes, um, because I started a bit the time a bit early. So I'll go to the next slide here. So here I have 20 minutes to complete this test. Do not rush. If you cannot finish the test in the time, move on to the next section. So I initially finished the version B. I'll go with version A. So before that, let's set the timer. Let's clear it to 30 minutes. Sorry, I'll just refresh it. 0. Yeah. So this is the timer for 30 minutes. I'll go with test A now. And yes, next. Alright, so I again have some cell diagram, and I want the first question I'm asked, label G represents several proteins that assist with cell division, protection and maintenance of cell shape. What do these proteins comprise? So this is G. This doesn't look like, so there are 3 things. I'll go with cytoskeleton because this is something I remember. I'm not sure what these 3 different types of structures are. Right, so label F represents this, which is responsible for F. F looks like centrioles. Sorry, but this looks like microtubules. The centrioles were a bit more rounder, I guess. The cell division is responsible for nucleus. So the organelle represented by label D is mainly found in and dash. Your answer should be the names of the organelles. Separate your answer. The name of the organelles mainly found in label D. It seems this is ribosomes. Yeah, this is found in it's mostly found in cytoplasm, I guess. And there was this smooth surface and a rough surface. It's called endometrium. Endoplasmic reticulum. That's to the rough one, not the smooth one, I guess. Main function represented by label L is the Golgi apparatus has looked. The diagram has changed drastically. So the options are K and L. It was a bit more wavy structure, so I'll go with K. K, lipid synthesis. I'll go with receiving and sending. So I'm sure it's K. The organelle the organelle label C is present in the organelle label. It's the nucleons. There's a structure inside the nucleus. I think it's present in both. The organelle label will be in higher numbers in muscle cells compared to fat cells. So organelle labeled. This looks like animal cell, not the plant cell that I'm sure of. I'll go with H because that looks like mitochondria. Just as amino acid is a monomer of protein, dash is a monomer of DNA. Number of 14, a dash is a, I guess, phospholipid as nucleotide is nucleoside is. These are the regions where it's present. I'll go with pentose because this is the name I remember. A DNA monomer is called a nucleotide. Yeah, so I'm sorry, but I have to change my answer. A DNA monomer is called a nucleotide. It is made up of 3 parts. Nitrogen is base. Which part? The sulfate group is correct. A deoxyribose, of course, it has nitrogen is base, a deoxyribose sugar, and there's another part. I remember it has an extra OH group and RNA. It's a sulfate. No, it's not a sulfate group. Sorry. This is more stable. I'm sorry, I don't remember this third part. It was a phosphate group. Yeah, so there was a phosphate group instead of a sulfate group. Yeah. And therefore, less reactive. Justify. It has one less hydroxyl group, which I'll call it hydroxyl group. Well, I'll not be able to answer this at all, I'm pretty sure, but let's go. Use this diagram to answer the following 7 questions. According to the strandedness of this molecule, what type of nucleic acid is this? This is delta CCA. Delta was TC. Okay, so I'll go with DNA. What are the type of bases? What type of a U? So I guess the above one shows the strandedness. I'll go with RNA because what is paradoxical about the image, I am not sure. So it has cytosine. I'll go with 2C. 2 cytosine. According to your answer to 11, what does T? I think delta is T. This is epsilon is A, if I remember it. Delta and 3 and 8. What these questions repeated? What bases do? Use the following diagram along with the previous to answer the following 2 questions. This is a sugar from previously discussed nucleus. There is a hydroxyl bond at 2 and 235. 235. Okay, what type of nucleus? This looks like, let's count. I think this that either, I'm not sure about what DNTP is, but the conclusion, DNA can sometimes include, that's absurd because it will make the nuclear DNA unstable. The backbone of DNA can sometimes include this type of sugar. It matters. RNA can sometimes be found. I'll go with this one because it can be, but it's not that stable. The ordinary flow of information in a cell is, this is, I know, DNA, RNA, protein. The process that takes place at the first arrow is transcription and translation, and the process at the second, this is the last thing I read. Which statement about protein is correct? An alpha helix is indicative, a beta sheet is indicative of the structure. So multiple proteins interactive with the quaternary structure. I'll go with the A1. I'm not sure about this one. A DNA template strand consists of 5 sequences. What is the complementary mRNA? I can't answer that. I'll go with C. This is compulsory. Your mRNA strand of choice must be reverse. Must be written complementary. What protein sequence does mRNA code for? Look at each protein. 3. RNA is made up of U adenine, UAC, adenine. Sorry. Yeah, they're separated by an hyphen. Each protein. Let's go with A. Therefore, I think this answer is incorrect, but that's the only thing I remember. A point mutation causes an amino acid to change to a stop codon. I don't know what stop codon is. What is the effect on the resulting polypeptide? So the amino acid is changed. I'll go with this. The second point mutation causes that stop codon to change into a that is different from the what is this? Silent, I guess, but these two completely guess works. Right, so if we're going to solve, let's stop the timer now. Okay, so let's view this score. Oh, sorry, this is wrong. Okay, correct answer is I was correct, but I mentioned the order wrong. Maybe. Okay, this matters here. Yeah, this is correct. Yes, justify your answer. So I'll treat this as correct, but it's more like according to this trend, this is you. Okay. Represent indicative of DNA. Represent of uracil RNA results into physical. That's pretty standard. Oh, okay. So this is going to be a conclusion. Sometimes it's double stranded. Ternary flow, this is correct. The statement about proteins is correct. Beta sheet is indicator of the secondary structure. I knew this is going to be from 5 to 3. Okay, so what protein? This I could never have imagined this answer ever. And done. Let's see what's asked. Sorry, it's asked next. Yeah, so okay, I've lost my Safari page. Yeah. So I have 20 minutes. Take a 5 minutes break. Okay. This is done. I have done. Please complete both of the surveys. Just why am I able to only see one survey here? So copy to. So didn't use a model. Next. It is pretty high demanding. Physically demanding, I would say I had to scroll a lot, so I'll give it a 9. How hard? It was pretty rushed, but yeah, it was pretty rushed. I'll give it 10 because I was not able to finish. How successful? I'll score myself as 7. I did better. How hard did you try? I was irritated by the amount of searching I had to do, so I'll give it a 5. Yeah. Okay, so this is done. So I think I'm done with this recording. So thanks.